and longevity. And the goal here is to have first information coming from Anton Kulaga and other people who are online that we try to join at the moment, no, who are already listening online at the moment, but and that you will probably see after. And the main idea here is first to have information about AI and longevity, and after that to have a kind of a very large brainstorming about what could be improved, and also what is great in what Anton and his friends are doing, and what could be done even outside of ChatGPT longevity to go further concerning what is the goal of many of you probably here, and certainly for me, and certainly for Anton and for me, longevity, a longer and healthier life for everybody. So now, if everything is working, we will have we will have Anton speaking. Thank. Yep. Yes. I have this. Yep. It's yeah, but yeah, you can continue already. Normally, it's okay. You have to.
Yeah. So hello everybody. I am Anton Kulaga. So I am a member of Systems Biology of Aging Group. Also, I recently joined the IBIMA group that does aging research in Rostock, and I am also life extensionist, transhumanist, and involved in many longevity and open source projects. So today we will try to kill like 2 dragons. So the first thing is machine learning models that are the most important for aging. So it's called aging clocks. So probably most of you already at least saw some presentations or read some articles about aging clocks, and some of you probably already measured methylation or blood clocks. Actually, I'm actually curious, could those of you who ever measured any type of aging clock, can be blood, can be methylation, raise their hands? So Sven, I think you did, no?
No, I didn't.
Oh, okay. So just is it only me? So no methylation, no aging clocks, nobody tried it? Okay, so today we'll actually fix it because I plan to make it a little bit more practical, and actually we will try to measure at least 2 aging clocks, and in the end probably most of you will be able to raise your hands, and also you will understand what actually to do with those aging biomarkers. And the second dragon that I want to address is that everybody knows about large language models, and probably many of you already use ChatGPT, Llama, or maybe some other language model. And of course, there is a lot of excitement how we can use it for aging research and other transhumanist causes. Actually, question, who of you ever used ChatGPT or any other language models? Okay, so we have majority there. Who of you has ChatGPT 4 access? Okay, so we have 1, 2, 3, 4, 5, 6 people. Great. So today we will actually try to use some custom ChatGPTs that we made for life extension courses, and we will try to make your own one. Yep.
Can you share your screen on Zoom?
Oh, okay. I will. It's not shared on Zoom.
Not at the. So you.
Okay. Thank you. Okay. So I will share my desktop.
And maybe for the sound.
I don't know. Nobody gave me a microphone.
No. It's I mean the mic. Yeah.
Microphone on my computer.
Yeah. So the.
Okay. Great. Then let's go further.
Your microphone is not on?
Yeah. Join. I just discovered that I was not joined with audio. Yeah, so just for online visitors, I will repeat that today we will get some theory, but we've also try to compute some of our aging clocks, and we'll also play with custom ChatGPTs designed for life extension. Yeah, something that can be useful is that hello. Oh, great! Now I have the microphone. Yeah. So just I give some links in advance. Maybe actually I can give also a link to the presentation for the Zoom attendees, so they can they will have easier access towards the links. So where is the chat? Yeah, so in the chat, I will also give link to the presentation. And okay, great. So first, because I noticed that most of you didn't measure any aging clocks, I think maybe I should talk a little bit about it. So, I mean, probably all of us want to solve aging and probably prefer some like second scenarios that when you will get older, we will be able to repair most of our damage and feel young again. And of course, if we talk about aging, that the main way how we detect aging is that it's increase in fragility and vulnerability and the deterioration with time that leads to death. And there is actually like other than just measuring death, there is also the way to measure how number of our diseases increases. And here you can see an exponential increase of different diseases with time. And if you just cure age-related diseases but not aging, you will probably die. You like solve some diseases, but you will die from others because all of them go exponentially. And if we look into aging, so there are different theories, and the most popular ones are about damage accumulation in different parts of our organisms, and on cellular level, on tissue level, on level of genetics, epigenetics, and overall, the idea that the goals that we want to achieve is that we want to figure out how we can fix those damages. And as you can see, so this is like our performance level, and with the interventions we want to fix the damage. And there is also a saying, live long enough to live forever, because with time more new discoveries appear that can even more reverse our aging. And here just was a reference current records achieved on model organisms, like 10 times on worms, and with a combination of genetics and also diet. You can see that 100% extension was available on mice. Unfortunately, the more developed the organism, the harder it is to increase lifespan, because many things that are wrong in short-living animals. Because, for example, for a mouse, the highest chance to die is from cat and not from aging, and that's why they don't optimize for living longer, because they optimize for multiplying faster and for running away faster from cats. And that's why many things that work on mice, they maybe not working on longer living animals like humans, because humans optimize it. And if we talk about measuring aging, so the main problem is that for short-living animals like mouse, you can just measure how much it lives, and you make an intervention like a drug or genetic therapy. You wait several years and you know the results. But for humans, we cannot wait 100 years to check that some drug is working, and we want to see some indicators that actually it is rejuvenating us. And for this, there are different types of biomarkers, and usually we look into two types of them. It's functional and physiological. So functional is how actually well we feel, like how is our memory doing, how is our performance doing, how good are we at sports, and so on, how fast we recover, and so on. And for physiological, we usually measure different methylation, blood, cellular parameters. And you can also see this actually chart that so there is a trade-off between how accurate you can assess aging and how actionable and explainable it can be. Because the major problem, for example, when you measure methylation, it's pretty accurate markers that many people measure. You kind of know if what's your expected age. For me, I think it's several years younger than I am now, but you don't know what to do with it, because you cannot understand what is bad and what is good. And for our personal usage, the most important is actually we have to make changes on our life based on it. In terms of actually biomarkers, I was co-author in a review paper where we also had another classification, like molecular and systemic and physical capacity biomarkers. Let's look into actually how our functions degrade with age. So here is a very well-known a marker called muscle atrophy. So here you see actually how records change, like 100 meters run, 400 meters run, jumps, high jumps, and so on. So you clearly see the degradation. And also in terms of endurance, so here is, by the way, photo of the oldest marathon runner. So 104 years old. And here, like how the records change. So actually for marathons, surprisingly, 30-something is the best time to achieve results. And unfortunately, so like 3 months ago, I ran a marathon in Bucharest, and unfortunately, I'm like 70 years old, based on my speed and result. So I definitely have to improve on this to feel younger. And we also have similar things with memory. And it's actually kind of surprising is that most of our abilities go down, and probably, but there is like one exception is our ability to use to know our world and to use our words. So we degrade on everything other than blah 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 blah. That's why many politicians are that old, because they develop their skills through lifetime. And actually it's increasing. I think if Biden will win, he will be like the oldest president in the next term. And yeah, so these are examples of functional biomarkers. There are like many of them, so you can, and they are relatively simple to measure. Like, for example, grip strength is surprisingly correlated well with aging, and you just need a very cheap device to actually measure. And same with most of the exercises, you can just see how your performance improves. The issue is that actually, for example, for me, grip strength would not be a good biomarker because I'm doing climbing, and obviously my grip is getting stronger than wild type people. So that's why it's also important to look into physiological biomarkers, and they're actually, it's kind of interesting because here we don't have like obvious indicators like how fast I run, how high I jump, and we have to somehow interpret. So we take measurements like blood, methylation, and other types of biomarkers from young and old people, and we train machine learning models. So most of so-called aging clocks is just machine learning models that predict how old are you, and when you evaluate how good are they, so there are like three most popular indicators. One is so it's actually pretty simple, mean average error. So on average, when it predicts how old are you, how many years it makes as a mistake. So if mean average error is like 5 years, so it is usually it is mistaken around like 5 years plus minus. Then people also measure R squared, that well, is a bit more reliable actually in many cases, and also Pearson correlation, how well it correlates with aging. There is one important aspect that even though these metrics are used pretty much for most of machine learning models, with aging, we actually often don't know when, for example, it shows me that I am like 4 years, for example, for methylation, I think I am 3 or 4 years younger than my chronological age. But for glycan age, I am 12 years older. And you have to understand that in some cases it can be because the model makes a mistake, but in some cases, if it is connected to biological aging, it actually may mean that your health is not doing well and you have to do something about it. And when you make an aging clock, you have to validate it with people. For example, usually they look into cancer survivor and people with accelerated aging. For them, they know for sure that those guys, they should should like age faster. So if the model shows higher biological age than chronological for faster aging group and vice versa for longer living aging group, then probably a big part of its inaccuracies is not a bug but the feature. And another way, if you have longitudinal data, and I mentioned in the past, you saw the chart that instances of diseases, they increase with time. So if you can see that aging clocks for people that get diseases and get deterioration of functional markers, and if it's a long-term cohort, maybe like 20 years, if they are doing better than average, and if the aging clock shows that they're younger than their expected chronological age, probably this clock has some meaning. And if we look into the clock, so there are, depending on different markers, there are different types of clocks. The easiest one, and a bit later today, I will give a word to my colleague Nick Hill, who will actually one question. Has any of you, for example, on your email, have any like blood test results? Can be like PDFs or anything. Is it possible for any of you to find like regular blood test results, like on your emails? And so for Sven for sure. And okay, so we'll see if for those who will find, we can actually compute how the aging clock. And the other aging clocks that actually we will try today, it's facial aging clock, because it's super non-invasive. You have to just make a picture of you, and the clock is trained to predict your perceived age, and usually it correlates a lot with skin aging. Also, it has also its downturns. So, for example, I can find some rejuvenating light conditions when the clocks make mistakes in one direction, but not in another. And for these clocks, actually, you see that if you look into the metrics, that like 5 years mistake, some cases 3 years mistake. So these clocks can be pretty accurate, and they are very easy to measure. And actually, let's actually measure. Guys, can you open? So I give a QR link. Can you check if it works? Or you can open the link. I accidentally muted myself. Yeah, so first, like few words how it works. So this is not just a regular it's not just a regular aging clock, like facial aging clock. They also use so-called places smography signal capture. So they do a picture, but they also do a video. And there, when you have a skin and you have some blood flow, based on like how blood goes through your veins, you have changes in luminosity here. It's very subtle changes. So with eye, only if you are like super trained people, you will be able to actually understand the heart rate of another person. Some people actually can do it after extensive training, but most of us cannot. But with proper machine learning models, actually you can infer quite a lot of it, like heart rate variability, even like blood oxygen saturation and respiration rate. And this model that we will try now. So let's see, maybe I can, so I have scan results. And so from scan results, you see that on this model, they give like vital information, they give some cardiac load, vascular capacity, and they also give also some information about blood, not super accurate, but still useful. And if you look into the list, also age estimations. For example, here, where is my, let me see, where is my age? Mental estimated. So I also give like some information about like my weight, so they have some a bit more information. Facial skin age. So I am 36, actually 2 years younger than I kind of like it. Maybe. And you can also do it like on a website. So you can do scan. So guys, are you trying to make a scan of you with your phone, maybe? Anybody? So yeah, so let's try it, because in such case you will at least say that you measured at least one aging clock. Loading. Okay. Okay, center your face. I hope the internet will allow. Actually, I don't know, maybe if my camera is shared with Zoom, maybe there can be issues, because I centered. Is it working for you guys? I can do it like with my phone, but I don't know why. So I centered my face. Okay, now actually maybe it was just a caching issue. So the idea is that you put your face, then you look to when it will show enough stars, and then you will capture the video. That's, I mean, I have a microphone that I will like right now record without it. Okay, I think that the quality of lighting is not really good here. So the light is on everywhere, right? Because it doesn't looks like 2 stars. Too far. Yeah, maybe. Let's see if. Okay, it looks really. No, maybe your like light is trembling. That's why it. But I did it measurements. Yeah, so looks like with this lighting, it's not any. But I did measurements just before this presentation. Question: any of you managed to like get normal measurement? Okay, so it finished for you. So can you check what's your age there? And in the app it's 38, and your real age is, wow, you look really good. But you have to measure it under different light conditions, because how many stars for when you measure it? 4. It's actually pretty good. Usually I measure during 5, and specifically like this. So for this aging clock, it's actually done by the team of Andrei Patescu. He is quite active in Romanian transhumanist community, and it's pretty like new one, and they only had 3,000 samples, and they're like collecting to improve the models, and so far it's a bit variable with the light conditions. That's why for this clock I do like multiple measurements with 5, but at least like you got some results, right? Anybody else got results with the 4? No? The age, okay. So you have, wow, I think actually my laptop froze. 24. Okay. Who else tried? Charlotta? What's your, well, how is it? Are you younger or older? Ah, you see? So yeah, so you can say it's a good clock. Well, actually, it looks like my computer froze for some reason. Maybe I overloaded it. Oh, it's really sad, because maybe I should I don't know, is it streamed to Zoom? Because for me it feels totally frozen. Yeah.
I'm just wondering about, is it really just using information on the scan, or is it technology
types of clocks, and they also depend on different features. For example, for the clocks that are like glycanage, it depends on immune status, and as I have like thyroiditis, it gave me a really high range that I am having. But yes, so I am back to Zoom, and yeah, sorry for the Zoom audience. I like my computer froze for some reason, and I had to reboot. So going, yeah, by the way, I think I skipped this part. So this, okay. Yeah. And yes, so coming back, by the way, I skipped, I think last time this part. So here I also wanted to reflect the things that we talk about, how our lifespan changes, like in the very, very past, we had really short medium lifespan, like 50 or 60, and now we are having way longer. So what changed during the last like century is that you can see the curve. So 100% is like in the beginning, how many people live, and then they died when they reach certain age. And most of the, so when you actually change the lifespan, and when you do lifespan tests on mice or other animals, it can, if our ideal solution is actually to scale it to the right, because we will increase both the maximum lifespan, like the longest living one, and the lifespan of everybody. So it will just move the curve to the right. But what actually mostly happened is that you can see that with age, what changed for humans is there was a little bit move to the right, but most of the movement it was increase in average lifespan, and because we learned how to cure diseases, and we got rid of child mortality, but unfortunately, maximum lifespan change only a small amount, and the idea of true aging intervention is actually increase it to the right. So yeah. But let's come back to the place where we stopped. Yeah, so here is a list, some list of facial aging clocks. And yeah, I did the demo. There is also clocks based on MRI, and they're actually really accurate and are very accurate also for brain aging. So you can see here like mean average error like 2 years. So they are imprecise like only on 2 years. And there are also clocks about movement, and it's also clocks, for example, Jira are developing them that you can get from just your wearable devices like this, for example, watch. And overall, the clocks that you can get from wearables is also called digital biomarkers, because it's mostly about processing digital data, and you can make a lot of measurements with them. And a few words about omics clocks. So those who don't know, actually, I'm curious. Who of you didn't hear about methylation or epigenetic clocks? Okay. So we have like only 5 persons who never heard about them. So that's why, like, simple analogy. If we look at our like organism, we have So genomics is our DNA, and genomics, at least to my research, it's around from 15 to 25, 30% of how long a human will live on average. Other factors are your lifestyle and so on, with exception of maximum longevity, that is way more heritable. Transcriptomics is like when we read like from a hard drive, read information into memory. We use like RNA like as a intermediate. It's like computer analogy is like RAM. And from RNA, we build proteins that do most of the work in our body. And for each types of this data, we have, so this is actually different types of sequencing. So everybody knows like DNA sequencing, but like those that take genes that are actually sections of our DNA, but you can see that there are a lot of others because we have different, so DNA is like are put on top of packed on top of histones, so we can sequence them. We can also have marks, methylation marks on DNA, we can sequence it, and so on and so forth. And here, if we look into those logs, so transcriptomic logs is based on RNAs, and so we actually, I also developed some of them. They are not super accurate, but they are easy to explain because you know which gene is more or less active. So not that accurate, but easier to understand what's happening. Then methylation plots, the most popular and the most one of the most accurate. So here we have methylation marks on your DNA, and in simple, it's if something is methylated, usually this DNA is less active, so we have less reading from this DNA, less production of RNA, and less proteins. I'm of course simplifying. And their methylation patterns, they change with age. Often we have so-called random drift, because methylation is the way, one of the ways how we, for example, differentiate cells from each other. Because if it is, for example, blood cell, it expresses hemoglobin and it's red, but you don't want to have hemoglobin expressed by neurons, muscles, and other cells. And that's why hemoglobin is not active, it's methylated and tightly packaged in other cells in blood, for example. And with time, we have like accumulation of random methylation, demethylation, and cells partially start losing their identity. So we get like some stuff that is expressed in wrong types of cells. So you can get accidental, for example, hemoglobin expression in neurons that you probably don't want. And we have millions of methylation points in the DNA, and people train the models. Usually it's a type of elastic net. So it's like elastic nets and different types of regression. So pretty simple models that pretty accurately show the aging phase. The problem is that it's so here you see, for example, just some of the blocks. There's like huge number of them, and the problem is that often those methylation changes, it's happening somewhere in between genes, and you have no idea what it is connected with. So when you look into changes with age, some of them are causal, and like if it changes, it causes aging to go faster. Some of them are just random accumulations that doesn't make you unhealthier. It just happens with time, and that's it. And some of them are adaptive changes to compensate for aging-related damages. And if you try to fix them, you actually hurt yourself. And with methylation clocks, it is way harder to actually understand what is what, and make changes to your make serious changes what you have to tweak, other than like general recommendations like exercise more, eat less, and so on. And people also check for different many clocks, and people even check how they correlate with each other. And you actually see that many of them, even though they are pretty accurate, but they don't really correlate well, surprisingly. And probably they are like connected with some different aspects of aging. And also on top of methylation clocks, there are also talks about proteins, talk about inflammation. Yeah, and here actually you can see how different clocks. So glycan H is mostly like glycan stuff on antibodies. Here you see immunological clocks. Here you see lipids like fats. Here you see metabolomics, and you see that here forwards anum is all methylation clocks. So bottom is methylation clocks, and you see that many of them don't really they correlate, but they don't correlate super well. And also something useful recently that Gladyshev lab in collaboration with several other labs, they made this BioLearn library and also a leaderboard to compare different clocks with each other, and also to let people compete who can make the more accurate aging clocks. And as I mentioned previously, that we always want not only just to understand how fast we age, but how we can tweak what we are doing to slow down our aging. So we want to understand the causal, what is causal, what is not, and also to get some actionables. And so it's not only, so aging clocks is not only like how accurate they predict your age and also biological age, but also what you can do with this. So there are like two types of models, like black box and white box. Black box is just, it shows like you are like 25 years old. Why? Well, you don't know. It just says it. You don't know how it made a decision. While white box models, it just explains that I looked into this feature, into that feature, and for example, your hemoglobin is bad, but maybe your T cell lymphocyte count is good, and you can understand how things contribute. And the issue here that you have to understand is that best models are usually something with neural networks that is extremely hard to explain. So the best things are less explainable. At the same time, we actually want to understand them. And for this reason, people, actually it was research of DARPA, like American agency for defense research. They made this concept of explainable AI. So here you see like comparisons that in this case, it says that this is a cat and no idea why. But in this case, it says that, oh, it has fur, it has whiskers, it has claws, and so forth. It is the cat. So when you get how it makes decision, you know what you can tweak and also what you can address with different drugs and other interventions. And the thing that is also interesting about AI is that the way how it makes decision can be very different from what humans do. So for example, here is an example of one pixel attack. The idea is that in some models, you can change only one pixel. That is the most important for the models in making decision. And we see that sheep turns into a car, horse turns into a frog, deer turns into the airplane, and into the dog. And another type of attack is adding some noise that you cannot even understand with your naked eye, but it can change panda into gibbon. And that's why for us it's and also many of these features, so usually humans will look like into some features like a nose, like whiskers, but in some cases there were like cases with machine learning models when there were security models, and they made a mistake when somebody was wearing a tie, and then he came without a tie, and it couldn't recognize him because it learns that this specific person always wears a tie. It was long ago. Now, of course, the models are much better, but sometimes it can make decisions on features that are not really important, but by chance they are predictive for the training set. And for this, we have actually the models that, so we have our black box model that is very accurate, and we make a separate model that can use this model as a tool and play with its inputs and outputs that tries to explain how this model behaves. So you take like some black box, hell knows how it works model, we play like give a different task, like see, okay, we give a cat, we give a dog, how it changes prediction, what if we will mix cat and dog features, how it will behave, and so on. And after some time of iterations, you can actually understand how decision is made. So there are like models I don't want to go into details of this. There is a whole lot of explanatory models that exist. For pictures, people made this heat map explanations when it highlights, like, for example, that why some type of like why decision, also that this vision, for example, is important, like in X-ray detection, so you know how it made the decisions. And you can see that actually like it's getting really popular. So it's like GitHub stars, and you see like a lot of methods. I personally, in my research, I used SHAP a lot. Yeah, so these are number over time. The Shapley value explanation is that they try to permutate different features and in a specific way, and then it shows how changing something shifts the prediction to higher or lower. So this is how it is used for aging research. So probably when Nikhil will talk about blood aging clocks, he will like to go in more details. But the idea is that what you want to know is not only that I got some age prediction, but here are like different like bond, total cholesterol, and other parameters. And by default, for example, we know that if like basic, but on like, you know, like average temperature in the room, so average prediction would be like 40 years. And then it looks like, okay, this band, it has this value. So from like average prediction of the population, these parameters that you have, it will add like three years to your age. So it will age you like 3.5 years. This will age you like this amount of time. So you can say that probably my band increases predictions towards higher age. Probably it's not good. I have to do something against it. While here you can see that triglycerides, and wow, they're probably typical to a person that is younger. That's why the model thinks that it decreases prediction of the model by -3 and 9 years. And with this type of explanations, you can get some actionables, what actually to do. So this type of explanation is called local explanation. Local because it explains specific prediction, like your own result. What is bad and what is good. There are also global explanations that explains for many predictions. These are examples of global explanations. It shows that you have some middle value for the population, like an average expected prediction of a model, and high value of the models, for example, are red, blue, are low, and they can actually, in some cases, high values of carbon dioxide of some feature can increase the prediction, but in some cases higher values can decrease it. So it's sometimes very non-linear. So this is an example of plots of global explanations. And here are examples that people are trying to apply this technique. I think this is for methylation clocks because they finally want to understand, like you have millions of methylation points, what they can talk about. Yeah, so in short, for like explainable AI, that's the key takeaway that we want not only to get accurate predictions, but we want to understand how it makes decisions, because we want to know what we can tweak and what we can target with interventions. And now I will give the word to my colleague Nikhil. So we'll see. Can I, Didier, can we kind of switch it so we will hear Nikhil, or how does it work? You're host. So, Nikhil, can you unmute yourself? Let's see if we hear.
Is my voice audible?
I don't hear him.
Is it audible right now?
We should change how this. Nikhil, can you say something?
Yeah, is my voice clear?
Yeah, and probably we should. Can you share your? You can open presentation and share, and
yeah, sounds good. Just a minute. I am starting to share.
Where is the mic?
Yeah, I guess it's clear, right? Cool. Hey guys, thanks for letting me speak here. So
Nikhil, wait a bit. We will see how we can. Do you have any speakers you can connect to laptop?
No, we don't have it, but normally I already done that with a micro, but I just have to increase
Locate where it is.
Yeah, I don't know where is it. Okay, can you try to speak?
Nikhil, can you say?
yeah. Can you hear me?
Now you have to connect your computer.
Yeah, we have to
Connect,
change
yeah.
the setting to the bank queue, the bank beamer. Now it's set
So
up.
I change what sound setting?
Yes.
Okay.
And then we
Okay, sound. So I put it to what, HDMI?
yes.
Okay. Okay.
Try to speak now.
Can you speak?
Is it better?
Yeah, it's better. It's okay.
That's perfect.
Okay, cool.
Thank you very much.
Make it. Mm-hmm.
Cool, then can I start now?
Yeah, you can.
Yeah, thank you. Thank you, everyone, for joining here. This is Nikhil. To briefly introduce myself, I am a machine learning researcher, and I work in building aging clocks and other LLMs for longevity and healthcare. I'm also a founder of this company called Helo, which also works in the similar areas. But today the focus is to understand aging clocks and LLMs. So I'll discuss more about that. And the other AI projects which are fully open source that we are doing to accelerate research in longevity and other areas as well broadly. But first, going to the demo of the blood aging clock, Anton has already described so many relevant things regarding this, but I just want to help you guys understand how we should efficiently use such a blood aging clock, because there are so many accuracies, so many numbers, and lots of variance and biases. And how do you come up with some actionable insight is of utmost importance here. So before going that, to briefly summarize, so the risk of each and every disease increases exponentially as you age. This is actually very central to building an aging clock, because your chronological age, which is based on your birthday based age, it keeps increasing every year, whether or not your personal risk of disease has increased. This graph is for an average person. For an average person, as their age goes from 40 to 80, the risk of diseases has exponentially increased, but it might not be true for you. So a good aging clock would not say that your age has increased unless your risk of diseases has increased. And it also works the vice versa. If in any given year, if your health deteriorates significantly for whatsoever reason, then your age in that one particular year has to accelerate way so much. And if any given year you took all precautions and you did your best with respect to health, your age has to slow down. So the goal of all of these aging clocks is to figure out on this curve, which is designed for an average person, where exactly do you lie at a personal level. And that cannot come from chronological age. That comes from you figuring out what is your biological age, so that you can put, suppose if your biological age is 25, then this is the graph. This is the points you need to look at in the y-axis. Yeah, and then so we already saw, because of one of the questions, some of the survival curves, which are also called as the Kaplan-Meier estimate curves. So I want to briefly describe here, not going too detailed into it, as to what's happening here. So this curve represents, on an average, at any given chronological age, for example, let's take 90, how much percentage of the cohort survives. Generally, more than 100, most people die, because this is on humans, and as of now, 100 plus is a very rare scenario. So you see that by the time everybody in the cohort reaches 100 years, the percentage survival probability is too less. And if you look at 80, then it's a bit high. But the point is, the green curve is for the people where their aging clock has predicted age acceleration. And the blue curve is for the people where the aging clock has predicted age deceleration. So you could see that you could use the aging clocks as a guide. And this is the proof that on an average, it helps you live healthier and longer. And the other important aspect of trying to understand how to slow down your aging, I mean, you could measure it through a clock, but again, it's another, but again, it should not just remain one another number that you have no clue what to do with, right? So in order to understand what this number correlates with, you could see that generally blood aging clocks like this that we're gonna see in a bit have very high correlations with various conditions that are generally not looked as healthy. For example, extreme obesity accelerates age by 0.7 years. And heavy alcohol consumption accelerates age by 2.5 years. And having previously smoked also accelerates it right now. And the important thing here to understand is this is like age acceleration per year. It's like some compound. It's like a compound interest when you take a loan from the bank, because if you have been heavily consuming alcohol, on an average, Again, it's very wrong to very precisely say what's going to happen to you, which is why we have to measure everything at a personal level. But on an average, your age keeps getting accelerated by this much as predicted by this endpoint every year, which is a very big compounding thing. But the good news is a lot of this is like adjustable. We just have to figure out what works for us. And if you did somehow manage to figure that out, and if you did reduce your biological age on an average by 3 years, then your money that you save in your life and your risk itself decreases, and the money that you save increases. So these are all like just some analysis on how to interpret the outcomes of the aging clock while you keep measuring it every now and then. Just measuring it once in a while might not be that useful. So now what I would suggest is a few things. One is that we could go to this link. You could all also scan this. This will take you to this website. The website looks like this. It might take like a bit to load, but I'll again put it over here for you guys to scan it so that you can measure your aging. Yeah, so hopefully if assuming everybody was able to scan this, so it goes to this page. This is what Anton had previously described about with an MAE of 4.5 and an R square of around 0.9. And we have trained this on 1.5 million plus records from all over the world. So what you could do here is you could enter your blood work from your PDFs. The units are already here. If you do not have any of the values, you could just leave it like that. It just considers a bit worse, not exactly the extreme worst case scenario, but the average worst scenario, just to be conservative with the value. So like I'll just change something, and so you could see your biological age. There's also a more accurate version which uses several more markers, and this will help you get even accurate prediction. But generally, screening these many markers is not necessary because the drop in accuracy is not much. So now I do realize that it's probably complicated for you guys to keep entering every number, but I would suggest that you save the link and you could later enter it, or yeah, or I have another idea, which is I don't know how feasible that is. If you really want to try, either you could enter all these things, or you could probably go to the Helome website. This is the Helome website. Here you could, if you're on Android or iOS, you could download these apps, and you could just upload your blood PDF report, and it will automatically parse all the results, and it would show you the values as well, the age analysis as well. Or you could also just without going to the app, just go through the online age calculator over here. But then if you go via this, it'll cost you $10. But going via the app route or going via this is like as of now completely free. If you prefer to go via the apps, then I should actually get this link. Yeah, you could scan this link. This will take you to the website, and you can put your email or download the apps later. whenever you have the PDF reports accessible with you, so you could keep uploading and figuring out the longitudinal analysis of how your age is changing, number one, and also how various of your day-to-day habits contribute to it. You could connect your variables and see which of the habits has the highest impact. For example, if you do consume alcohol, you would try to figure out if alcohol has any causal impact on the aging clock standpoint. And these causations are generally measured through, again, correlations and graphical models. I don't know how many of you guys have blood work right now, so you could either go through the app or you could change here. But what I would love all of you to do is, no matter which method you use, try to play with the parameters, right? If your creatine phosphokinase is currently 300, and if your age is so and so, okay, let's see what is the age first. This person's age right now is like 52. Let me 31. I am going to change the creatinine phosphokinase to 500. And so the age now has changed. So the thing is, looking at each marker individually is something that a doctor would do. You know, like each of these markers has a range that is agreed upon as something called healthy range. But when it comes to measuring the biological age, let us say if your red blood cell count is like way off the extremes, it may not necessarily mean that your biological age is going to be too bad either, because the aging clock is made through multiple complex trees and other compression algorithms. So it is always a mixture of several factors. So just because one or the other markers may have been extremely elevated, sometimes it doesn't really mean that it has a causal impact on the aging. It may still have some causal impact on some health conditions, which we do try to correct for. But our goal is to measure the aging that is not because of some temporary local disease.
Nikhil, we are a bit running a bit out of time because of technical issues. Can you like go quickly to digital twins, and we have to like have time for LLMs?
Yeah, sounds good. So yeah, I hope all of you guys got to try it out. And also, since all these tools are free, I would love for all you to figure out what has causal impact on your aging. And going back, and then going forward to the biggest issue with like AI and longevity is, so once you do figure out what impacts your aging, the biggest question is, okay, how do I find out a drug for it, right? There are so many billions of molecules that you could study, but the amount of molecules that we humans have studied so far is this. Just it's a tiny dot in that. And the another definition of anti-progression is that the cost to develop a drug increases exponentially, and also the number of drugs that are being developed has been decreasing, and in the sense that the life expectancy increase has been stunned. So generally, if something is better, if the technology becomes better, the cost has to decrease. But here the cost keeps increasing because of various sets of issues, which we are not really gonna go into here. But the biggest question is, can we use artificial intelligence to alleviate some of these problems where we could decrease the cost and improve the drug discovery process? So, but the idea is to use AI agents, because each AI agent is literally it's an algorithm sitting in computer. So it works at the speed of the computer's clock speed, which is around 10^4 operations per second or even more. We cannot have more than like 1 to 2 thoughts per minute, but an AI agent operates at like 10,000 times faster than us, and that too while we are sleeping. So the goal is to somehow make use of all of this existing technology, right? But you can't just deploy an agent with a goal from LLM, because then these LLM agents would it is a possibility that it optimizes for some stupid incentives, and it defies our purpose of even doing all of this. So what we have come up with is this very broad framework called the Digital Twin AI Agents Framework, where our goal is to not create a random AI agents, but for helping each one of us to clone ourselves into the digital world, so that each one of our goals are truly measured and interacted with each other. So I'll just skip. The idea of making the digital twin says that in future, when everything actually works properly, we could also do decentralized trials. And it also has other benefits in terms of other transhumanist goals in the future. So the way to do that is, it's a 3-step framework. The first step is to learn, which is where you create a digital twin where you put all of your data into it, including your health information. Number 2 is the planner agents, which plan on what are the tasks that needs to be done or interacted with. And number 3 is actor agents. Now, Longevity GPT that we're gonna talk about in a bit is just one of the actor agents. It like you give a question, it answers, and it cites every line in the answer. It's just one step of this entire graph of agents that we will have to sooner or later deploy, that will give us solutions at a much richer pace. And this will also be helpful for Longevity GPT. The way we take the data for Longevity GPT is just health papers, no personal information, but all of the data goes and then it gets stored as vector database, and then the digital twin safe would be able to communicate it with the actor agent. I will, since all of you have the link to the presentation, you can all go through it. Due to lack of time, I would suggest that you all can scan this link. It will take you to the GitHub repository with many more details about this entire open source project. And you could let us know if you would like to contribute here. I guess I can then give it to Anton.
Okay. Thank you. Thank you very much, Nikhil. Yeah, now let's go to our second. So I hope that you got some. Yeah. So there is an issue with microphone. I have what? Okay, I will share it with okay, I am sharing it now. Okay, so I hope now that you have a bit better understanding of how aging clocks work, and what are like the major challenges and issues, and how people try to make them more clear and explainable. And now we will talk about our second major topic, is how to utilize huge advancements in large language models for our longevity and transhumanist causes. And so I will be talking about the projects that Nikhil, and many others are involved in, called Longevity GPT and Longevity Genie. So you know that this year previous year we have this huge growth of large language models, and here is a well-known chart that tells how many days you need to reach to 100 million users, and you saw that ChatGPT just needed several days, way less than any of other large services. It's a bit outdated, but you can see how many large models are. And one actually of the things that I was starting doing when ChatGPT emerged. So you'll probably see this so on my t-shirt I have like my personal genomics results here, and here I give like examples. So this is actually my personal genomic results from another open source project I'm involved in, just DNA-seq. So you can upload your genome and get longevity variants. So when ChatGPT came out, I was actually curious if it can explain anything from my personal health results. And what I discovered is that actually, if you ask what is the longevity effect, so this is RS something, it's like RSID, it's identificator of mutation. So if you have mutation or some known, we also call it genetic variation, like you have one DNA letter in a specific position like T or G, typically this position, it has RSID. And you ask, when you sequence yourself, you know how it is called. So you ask, what is the longevity effect of this mutation? And surprisingly, ChatGPT, by that time, and actually even now, don't really know most of the information about genetics, with exception of really popular genes like ApoE and so on, but something less well popular, it cannot make answers. And it is also valid for many other health and science related questions, even though it gets way better. It also sometimes makes things up. You probably heard this word hallucination. Things got better with time. So if you're using ChatGPT 4, you notice that it hallucinates way less. And also it makes mistakes in reasoning a lot, because surprisingly large language models are not that good in math and logic, and sometimes they just run, they generate code to answer math and logic questions. And there was this hackathon at Zuzalu where we actually wanted to teach ChatGPT to answer better about age-related questions. And by that time, the problems were like hallucinations, not knowledge of longevity-related data sets. Now ChatGPT can actually use Google search to Google things about topics, but it's kind of general search. So it still often cannot find really useful things. And also, one thing that Nikhil already mentioned about digital twins is that ideally you want to have like a personal agent that you can tell all your health data to it, and it can give like personalized recommendation, knowing your preferences, knowing like your lifestyle, what you like, what you don't, and so on. So typically we started the simplest approach instead of because it's really hard to like retrain the really large model, and it's also cost inefficient. So we just integrated it with a bunch of so we tuned its prompts and integrated it with a bunch of data sources. So here is just an example of like typical AI lifecycle. Yeah, so here you can see that you have LLM, and you also have some code what's called orchestration libraries that just tells LLM some additional information, like you, for example, copy, paste, or upload documents to it. You can also do it automatically with software that can do it with databases. And we used genetic questions to actually test it. So here you see a general answer about, actually it was really nice set of positive, you see it's green mutations in FOXO genes, and ChatGPT actually knew nothing about it. Usually, if it doesn't know much, it is like a bad student. I will tell everything I know about this gene in general, something about biology, just not to keep silence. So you see, like most of it is pretty useless. And by that time, I already, so you saw Nikhil, and actually even before the hackathon, he did a project called Longevity GPT that he briefly mentioned, but you can actually just put on Google Longevity GPT. It makes search in research literature with integration with Semantic Scholar. It's a platform for like accessing the research literature programmatically. And you can see that it actually gives way better answers. It's still not ideal, less than we had in specialized databases, and you can actually test it yourself. And the most important, it gives references from where it's to, because ChatGPT often knows something, but doesn't have idea from where it knows it. So you cannot actually verify the sources. Maybe it's, I don't know, some random posts or anything. And yeah, so we decided that, okay, it's great to have Longevity GPT. Of course, we integrated with Nikhil as well, but we decided to connect it with specific databases and other resources, like, having a Swiss knife that connects LLM with all types of biodata, all types of databases. And we called it Longevity Genie, like a genie that grants wishes. And that's how the project was born. Later, so it was born at Hackathon. We got a little bit from VitaDAO, and also we got later in autumn, we had some support from this very nice organization called HILS. And what we actually achieved, so it's you can open the website. So on the website, we actually used on ChatGPT, they have a new feature that you can make customized ChatGPT chats and integrate it with your databases and applications. And you can just use regular ChatGPT interfaces. So far, we developed a bunch of agents. So we have the easiest for you to actually test it. I see like several of you actually have Chat GPT 4 subscriptions, because unfortunately it's Chat GPT 4 is required. Is actually through like the interface that you are used to. We also have GitHub organization, and we also have versions like that are standalone. And also you can always use Longevity GPT, that you can open it at the website and also use it. The issue is that when you use it like standalone sources, we have to pay for API calls. And also the interface probably is not that you're commonly used to. So here you see on the website, you have several like dedicated agents for different databases. So I will go through them a little bit later. We also had a Telegram bot that is used to answer questions. And so I will give words to my colleagues to actually show you how everything works and so on. But just as an example of the answers, you can see that my question about FOXO gene, when it addressed the databases, it gave actually a lot of detailed answer and also gave exact sources. And we did the technique when we use additional sources, it is called retrieval augmented generation. And the idea is actually just a short theoretical preview. So when you're using like large language models, you actually, it gets the text and then it generates the next token that usually work, but not always. And so it got like this text, it generated tokens, then it gives this as input, it generates next token, and so on. And the idea is that at some point, what you can get, like you can tell the, you can actually copy paste additional information from databases, like you, for example, do manually on ChatGPT. Here is my data, please analyze it. But you do it like automatically when, it can take it into account. And you can also teach large language models to actually ask for this information. So when it generates an answer, please call a database with genes, then it calls this program and it extends the text with the information from database, something that you actually do manually when you just upload information in your ChatGPT chat. And that's why you can get better answers. Another important thing is that for large language models, when it deals with words, it actually turns them into mathematical vectors. And it's reasons about them like points in a large space. So here, for example, I give simplified illustration like 3D. And you can see that there are like motions like banana, apples, and those that are close, they are usually closer together. And you can also use this feature to do like search, not specifically search by like keywords, but also search by notions. So like we can like search what is around cat, and you will also get like tigers, lions, and so on. This is called semantic search that this is also actively using. And actually the idea of this semantic search that you have this called embedding like representation in a vector form, and you can use it to search like in whole areas. And actually there is a whole specific models for embedding, and ChatGPT is actually not the best one. They use like ADA model, and their one is not the best one. So we are also using both calling databases and semantic search. And we indexed like 80,000 papers when it searches for the answer. And we also implemented like I think more than five databases when it tries to answer user questions. So here also like examples of some of the results that we get that it can explain from databases who lives longer, cats and dogs. And while ChatGPT actually is not that good into it, can also explain better in terms of genes. And it can also use different useful aging related tools. So I will not get into like details how we implemented agents. We don't have time for this. But I will actually go maybe closer closer to some of our new results and how we actually integrated with ChatGPT builder. And I think Maria, are you here?
Yes, hello everyone, I'm here.
Wonderful. So maybe we can show first how overall things work, and then we can go through some of the custom ChatGPTs that we built. Let me try to stop to give you like sharing access. Just a second. Yeah, I'm just it's a bit can you share? Because I lost the window somehow.
Yes, sure.
Typical.
You mean share the presentation or?
Share your screen, just the desktop, and maybe you can share like how custom GPTs work, so they will know.
Yes,
because only like 5 of them have.
So
Okay.
Can you see my screen, right? Okay, so my name is Maria Koval and I am a part of Longevity Genie team. And specifically, recently I was working on creating those agents or custom GPTs that Anton mentioned. And the first one is a demo of an age agent that is based on, here is a link on it for those who have a presentation can go and check it out. And inside this agent, we used this database NH from Hager. And now let's go directly to the agent and see how it works. So here you can see like we are in developer mode. So you can see all the configurations that we made for this agent. And in general, using this agent, because it's pretty simple, because we use just one file to retrieve information, it is a very simple one, so we can have a general understanding of how such agents work and how you can create similar ones if you want to, using your own databases or your own personal informations maybe. So specifically this one, agent will answer questions related to different species, how long they live, at what age they mature, males and females, and in general, everything related to animals. If you compare the results of answers of this agent to original vanilla answers of ChatGPT, they won't be very different. But still, we decided to show this agent to you just to show like a simple example of how everything works, a first one. So here is the name of it, description, just for people to understand what it does. And here is an instruction. That is an important part where you give instructions to ChatGPT of how he should act, what he should use in different cases, what he should do in different cases, and how to interact with the user. I will get back to it later to give you more details about it. Then we also have conversation starters, so you can give an examples of questions that users can ask, so they can understand how or what exactly they can expect from the agent. And here we also can add some different files, but there are lots of limitations that I will also tell about them later. But for now, we just have this one simple CSV file that contains all the information that I already mentioned. And there are also capabilities of agents. For example, they can web browsing, also create images like the one image that we have as an icon, and code interpreter. So this code interpreter should be enabled in case if you want to use files, because what is going on inside under the hood is that when you ask some question, agent just generates a code under the hood that just accesses information from file and gives it back to you. That's what's going on. And because each time it creates as a new script, it can lead to lots of errors. And this thing we can actually avoid. And also it will be, we will say about it later and different stuff. So going back to the instructions. So we describe that we give this specific file to the agent, so it knows that it exists. Then we give a very detailed information about how exactly the information is stored in this, like that is separated with commas, what exactly is the name of the columns, what the information is there. And as you may guess, there can be also some errors related to asking, for example, we want to know how long do cats live in general. And because this is a scientific data set, there will be only a scientific name of cat. So it won't be just cat, it will be something like domestic cat. In this case, we can try to avoid this problem by giving this kind of prompt to agent. So it will first search for scientific names in CSV and trying to find the corresponding one to the one that a user asked for just a cat. And it works most of the time, but not always. And there are also other ways to avoid it. But in this case, we should use already created code that I mentioned here. So as I said, because it creates new code for each question from a user, it can create a script that doesn't work properly. So in this case, we can create like some kind of API that will already contain a specific script that we already wrote that is already solid, and it won't be generated again from the scratch, and it won't contain any errors. So in this case, we can add some actions. And in this case, you will access APIs that already exist on our server, for example, and it will give you like more or less proper information. So in this case, we can avoid this problem. For this agent, that's it. That was just in case if it won't work for some reason. And we have another agent. It is Genetics Genie agent. This agent contains already lots of databases. For example, this Genet database, also longevity map database from Hager, and lots of databases that we created during our Just DNA sec project. And let's go to this agent. Here how it looks. It's already a little bit more complicated. Here we also give the same instructions of how it should answer to specific questions. And in this case, we added this action. So it is exactly what I was talking about. It is about adding API calls. So we can go here and see this is a schema of this API. And here are lots of different requests that we can get to get different information, lots of different methods to get different information. Because in this case, in case of this agent, we have lots of different data sets. And as for limitations of uploading files here, they shouldn't be very large. And sometimes you know that some genetic databases or in general databases can be very huge. So we have this limitation, so we can't upload big files. And another thing that if we upload, for example, 3 or more files, even if they are small, and we say like, for example, for this question, you use first file, for second type of questions, you use second file, and for third question, you use a third file, it won't really always follow your instruction and won't always access all the 3 data sets for each types of questions. So in case if we use APIs, we will also give the same instruction, like for this kind of question, access exactly this method, and so on and so on. And for some reason, ChatGPT will follow this instruction pretty well, never had any doubts, any problems with it. So that's how we can avoid these limitations with files. And as for examples of how it works and performs compared to ChatGPT, this is a question about mTOR gene and the answer from vanilla ChatGPT. It is a pretty generic one, but gives you a good overview of it. Not a bad answer. But as for getting an answer from our genetics GPT, it will provide you a little bit more concrete information, but pay attention to that. In the beginning of the conversation with this genetic genie, we provide the information of what kind of expertise we have in this field. So it can create like more advanced or less advanced answers depending on your level. So it will be also convenient for people who have not many expertise in this field. But in this case, it's expert level, so there is no much clarification, just solid facts, and mostly different kind of trials and papers. And if we want to find out some specific information for different variants, it can be also done here. For example, ChatGPT almost never answers well on such questions because it can give you a like an broad understanding also on some very known variants, but most of the time it's either wrong, or if not wrong, it doesn't provide you very detailed information on different tails and so on. And because we provide in Genetics GPT data set that contains specifically this information, it can be easily retrieved, and you will get a very detailed answer. In this case, it was already provided with information that the user is new to the topic, so it will give you some clarifications, what is S&P, a little bit more details, and not that advanced dry information, so it can be useful. And from my side, I think that's it. Thank you for your attention.
Okay, so I will also like show a bit how it is looks from ChatGPT perspective, because I mean, so here I mean, those of you who haven't like used Serious, so I will make it a bit larger for you to see. So you'll see like these customs ChatGPTs that you can make, and I have to share my screen. Okay. Yeah, constantly forget it. Okay, and here like so Maria, she focused like on how to make it, but I think it's important to understand. So you saw that she showed like conversation starters. So usually you want like give ideas what kind of questions your custom ChatGPT answers better. So here, for example, we ask about which genes variants are affected with specific disease. So let's say that we are experts, just because we want, and then what it does is that first, so you see that it takes actions. It means it accesses the additional data that you provided. Usually you have to allow it. And here you get like a lot of technical information that you know each variant, you know like what type of it, what it does. And here we are using the information from DiseaseNet databases. If, yeah, so you see like it gives a lot. And let's see what actually regular ChatGPT, yeah, so it's keep generating actually. And let's see like, okay, let's let it finish. And let's see what we will get in a regular ChatGPT for the same. So I'm copy pasting the same question. And so it says about genes, but it doesn't say any of the variations of this gene. And if you have like your own genome sequence, it's pretty useless information because, okay, I mean, it's some genes, but what I should look at. So you saw that it just said very general and pretty much useless information. It's only like 5 genes and no specific variants that you could look. While if you look into the answers that we got from Genetics GPT, so where it was Genetics Genie. Well, I think yeah. Or actually, I generated it again, but let's see. Here we have, hold on. Where is our history? Yeah, so here is your history. So you see it's like not 5, but actually it's 10 genes. And for each gene, it gives 3 or 4 specific, or sometimes 6 specific mutation. So it is the answers that is way more detailed. The way like what Maria showed you, that all of it is doing is accessing your it's making actions to call your REST APIs. And what is REST API? Is that like some application, that web application with data that you are running. You let me actually open it so you will see how it looks. EU, what how it is? No, this one is open search. Okay, it was longevity map. Longevity. Aging. So typically here, so what means like those actions is just you can call some methods on a server. And so large language model, it just, for example, if it is asked about disease, you instructed it that it should call methods on the servers that are there. So that's how it works. Works under the hood. And for genetic GPT, it can answer diseases, gene variants, and also specific pathways. We also have that an agent about clinical trials. So you can, for example, check if you are interested what is being done like in cancer, and it can give some either statistics, or you can answer about like specific trials. So let's see, it's analyzing. So this one is a bit, I think, slow. Yeah, so it takes some time to run the code. But if you like ask the same question of a regular ChatGPT, usually they just say something general and doesn't give a proper answer. Okay, let's wait. Okay, while we are waiting, I can also show the. So we have the one on literature, one on genetics, one on species longevity, and one on drugs. Newton Winter, are you with us?
Yeah.
Can you? Yeah. So Newton Winter is actually responsible for this agent. Maybe you can share screen and explain how it works, and maybe answer some questions, so other people than Sven will know whether they should take rapamycin. Use the share screen.
Do you see it?
Yeah. You can. You can show. Just open the agent.
Yeah. Well, initially we planned to show the reproduction of creation of an agent for you, so that you can follow as a hands-on workshop and recreate the drug agent yourself using the prompts and follow the challenges that arise. Well, Maria covered most of the internal details about how our agents are constructed. So since we're tight on schedule, I'll focus on covering the general ideas about what's to focus on when you create an agent that interacts with some kind of database. First, regarding recommendations of using drugs, the specific database, drug H is of limited use. Well, at least if you're not planning to give drugs to mice. In that case, it's pretty useful because the database focuses on animal models with which drugs are studied on, and it has a lot of information of longevity, pro-longevity and anti-longevity effects of different drugs, compounds, and additives for animals, excluding the models that are disease-prone, like cancer-prone mice and so on. So it's for healthy animals. And well, here are the examples of what ChatGPT generally gives us when asked about rapamycin effect on mice. Well, since we've talked mTOR pathway, here are the questions on rapamycin. Well, it follows the general pattern. It answers a lot of generic stuff with very little factual detail. Well, here it gives us a reference to an article in Nature without any specifics, and another query, basically same results, another numbers without reference, and once again a lot of generic wording. So what if we supply it with actual reference material to read on and answer in more detail? Well, first, this database DrugH allows downloading the CSV data. So you can supply it to the recent OpenAI addition to ChatGPT, custom GPT. Well, it's generally as Maria has shown. You upload this CSV, instructed how to address it with a prompt, and enable code interpreter, because this is how the GPT interacts with the CSV. It writes Python code based on a query and its instructions, and this accesses this file to get these answers. So actually, this gives pretty decent results. As you can see, the thing questions now have a lot of factual information with maximum lifespan increase data, average lifespan increase data, dosage, mice models which we used, and it even is capable of deriving sensible conclusions that outcomes based on different strains of mice, and it also gives proper references, which is important. Thus, references to the articles where these studies were conducted. So basically, since I have little time left, I'll focus on explaining the internals of the agent with highlighting main struggles when creating it. When you first access this custom GPT tool, you can use GPT Builder to give it initial description of what you want from the agent. When doing this, you need to focus on maintaining tone, instructing it to avoid speculation, instructing it to focus on the database scope only. Focusing the agent is very important because when it answers us, it has somewhat limited capacity to derive conclusions and well, making them thoughts. So we need to reduce the number of internal thoughts it does as much as possible, in the sense that we want to pre-compute the data for it that it should know before accessing the database. Basically, this starts with supplying it with the information what this database contains. In our case, we take the headers of the CSV file, and which can be also done with ChatGPT itself. We can ask it to upload it to the file and ask it to describe what the headers of the file are. So once we do that, the resulting description is now useful for the agent because it can know what the CSV file contains precisely. If you do not do this, what it will do initially, it will first access the CSV and spend some time analyzing it, and it will do it each time. You do not want that because it will reduce the amount of thought it will spend on answering your actual query. After supplying it with information about the headers of the file and what columns contain specifically, the other common problem with queries to databases like that are that ChatGPT can, to some extent, derive the scientific names from a common name, but for more general questions, like when you ask about fungi or I don't know, insects, instead of a specific species, it will fail to deliver your results because it will look for the word itself instead of trying to guess species from the database. So it can be addressed by adding some kind of lookup table so that in this agent, the drug age database was updated with common names of and taxonomy for species that are listed in the database. And this column common name is manually added to assist in searches. So we can look how it works on the example with Anjali. As we can see, it does not look through the strains or species columns directly because it's asked about a group of animals. And it looks through common name table with, just a second, with fungi, fungus. And as a result, it gets actual species and then performs the lookup on compounds for this species and answers actual questions asked. So let's have a look. Well, as we can see, it answers with the actual compounds for fungi, despite despite the fact that initially, without addition of this auxiliary column, it was not able to address this. Another option to help with translation of common names to Latin scientific names is usage of API, but it can also be circumvented with such somewhat simple technique. So basically, if you want to interact with database like Dragic,
Newton Winter, maybe we can go more technical with people who are like more interested. So I recommend like for those of you who want like to know details how you can make like a custom GPT on your own data, maybe you can contact us after. And one of the things that actually he showed is that if you don't make REST API, things can sometimes get complicated. Can you like in 2 words like sub-conclude your Dragic experience?
Well, despite the fact that without API you're somewhat limited in instruments, with doing some of the stuff like pre-computing some queries, giving auxiliary data and inserting it into the database, you can achieve pretty decent results actually with using pure CSV and code interpreter. So with prompt engineering, some amount of trial and error, and with adding auxiliary data to the database, you can achieve pretty decent functioning without API calls. Yeah, indeed, we prepared a hands-on workshop where we reproduce creating this particular agent from scratch, including the database modification, which is in turn done using ChatGPT itself. So you can supply 2 files to ChatGPT and get the modified file based on what you desire. So if you want, you can contact us and we can show you how it's done. We've prepared the prompts that I used for reproduction, but since we're out of time.
Okay, thank you very much. Yeah, so let's, okay, let me come back and conclude this. Yeah, so today we
so it will be so stopped during now exactly 22 minutes, and there is some coffee here, and that's it for 22 minutes. I hope to make general remarks and so on. And after that, the second part, after let's say 30 minutes, will be about really proposals of improvement. And Anton is here to answer technical questions, and I'm here to answer philosophical questions. I don't know. Yeah. So who is wanting to speak? The first, it's always difficult, so it will be Anders.
Yeah. Just when I thought that was safe. Yeah, wonderful to be here. And as you might have noticed, I arrived late, so I missed most of stuff. So I don't know anything about anything. But I'm from the philosophy department, so that's not going to stop me. Just a general point. Oh yes, but we in philosophy know we don't know anything. We're making that a big point. Here is an interesting thing I noticed when I saw the large language models working on these texts. As somebody in academia, it's not just hallucinations that really annoy me when large language models try to reason. They're very bad at constraints. They're very bad at remembering negations. They're basically going for the mood of the text, the style of the text, and making it as plausible as possible. But for actual use in science or business, it really matters if a sentence ends with a not or not. It really matters that early on, if you define a variable to be positive, it remains positive in the rest of the text. Adding those constraints is really important for actually making this useful for science. And that's not yet done, I think, in any of the language models. But this is something I think we can ally ourselves with a lot of the people in the AI world that say, this is really a killer application. This is what's needed for making it useful here. Then there's a second problem. That is, AI is wonderful, but you need to take it into a lab. And there is some issues about lab automation that maybe we can romanticize that. But we probably need to figure out a better way of tightening up the feedback loop between generating interesting ideas and leads and then doing the experiment and doing that so cheaply and efficiently that we get the rewards of AI. Okay, that was my voice.
Thank you, Anders. And now, so maybe there was a technical problem. So now, I suppose, can you write online? I suppose from here it's better. So the next one is supposed to come here. It's even more difficult. So who is going? No, joking. Who is going to make to ask a question, to make a remark, and so on? Please don't be shy. Yeah. You have to. It's better to come here because otherwise the people online can also ask a question that Nino will read again.
Okay, I guess my question is about utility or applicability of what we saw in the biomarker tools. Yeah, I forget which one was which, so I can't say which one I'm talking about specifically. But there was a tool where you put in a lot of different information from your blood work. And then there was a discussion of, okay, so now there should be drugs that you could use to affect each one of those, right? So for a user such as us, we don't know about the drugs, obviously. Something that would be interesting to us is to know something like, okay, so this aspect of how I live my life affects this component. So what should I eat, or should I do exercise? Which things that I can do that I know, that I understand, are going to affect which one of these markers, or these parts, these components, these parameters that get into that equation for your your age in that sense. That was one thought that I had. I also have a thought about LLMs, but we'll get to that later.
Or maybe, go ahead.
No.
Maybe the second thought. Yeah.
Oh, okay. Just one other thing that pops up. I can't say how it applies in this field. I'll say how it applies in my field. When you're trying to use LLMs as a way to understand, for example, in neuroscience, what you're seeing, to interpret what's there and to get a specific result. Say you're looking at a particular tissue and you want to get results about the neural circuits that are in there. It's very difficult to use an LLM directly, except if you're using it for very small components, because the LLM is trained to find things from a large corpus of data, and it will always tend to go towards those things that are statistically more likely, even if you can give it some noise to make it pop out of those main, solutions that it will find. So if you want to get something very specific, if you're looking for something unique, something that is not general, that is not just averaged over, say, all of the samples, but for that particular sample, you're going to have trouble. It's kind of like asking ChatGPT not to answer the question, how are, and then it will say you, but instead asking it to give you back, the rest of a poem that a specific poet made, something about the blue sky is effervescent in something. What's the likelihood that it's going to give you back the right ending for that if it never saw that particular poem? That's not likely. So this is one of the problems. You really have to understand when you're using an LLM beyond, say, just parsing literature and getting good results from the literature. I think it's very good for that. If you have a library of literature, trying to get something from there, it's helpful. But if you're trying to get specific results that are not in the main part of your statistic, but are towards some tail, then LLMs are not necessarily the right hammer for that nail. So just be very careful about your tools.
For this aspect, Anton, I think it's important that you give some explanation about this warm, cold, results that is going a bit outside of the most precise answer, because I think it's important to understand. And for you.
Well, it's technically, I think it's a data imbalance problems that, like in any machine learning, you have like some samples that are more prevalent and some are less prevalent, and that's why what are you showing?
There's another microphone in the laptop. If you can stand close to the laptop.
It's in the laptop because I thought I'm using this one. So if you fine-tune your model, you can, if you see that there is like a long tail of samples that are rare but important, maybe you can just show it more often during your training process to compensate for data imbalance.
Yeah, the case I'm talking about is one where you're dealing with a very specific sample. Let's say you're trying to reconstruct the circuits of this particular brain because you want to know why that mouse knows how to run through that maze. Then it's not a question of getting more samples of the same kind. You can only have that one sample. So that's why I'm just saying you need to know when to apply the tool. Sometimes it's the right tool, sometimes it's not the right tool.
Yeah, but the thing is that what you can do, for example, you have some specifics of this particular mouse, so you can generate the beginning that will fit in all parameters that you know specifically about those mice, and you can give probably general instructions what it should focus and not focus on. Of course, and also I don't know, I mean, is it like textual generation or you're using multimodal model? In your case, is it text or is it visual?
In this case, it was going to be the regeneration of circuits, so it's more of a visual thing, not textual, but you could describe it as text.
Oh, well, I mean, right now there is a trend to make it multimodal. So, I mean, there are some limitations. I totally agree there with you. So, also like from our experience, so there are some certain tasks when it makes mistakes, and you have to get, after you start using it, you get an intuition where it will perform better and where it will get problems. And you had a second question about biomarkers and actionables. So the thing is, there are like most of the actionables are like general, like you have to exercise, you have to avoid smoking. There are actionables about specific biomarkers if it's explainable, like what type of them, like what is connected for, like if you have low hemoglobin, what you should do, or if you have something else. Maybe so Nikhil has for blood, maybe Nikhil can say what's his experience for blood biomarkers. Nikhil, are you with us?
Yeah.
We actually don't hear him. Maybe.
Can you hear me?
You have Windows. I don't know how to configure it.
Oh, sorry. Can you hear me now?
Maybe it's better that he's writing, because it's not going to.
Okay. If you cannot configure, maybe we can address it later.
He's writing because it's online there. Yeah. Okay. Okay. Yeah. Sharp, but you have to. And then, if nobody is asking, I will give the word to myself.
Here we go. Yeah, just a bit of a clarification, because I thought that the two questions that you raised, Randall, were very intertwined. One of the things I thought of as a direction to take these LLMs into is to give personalized recommendations. And so you would have a sample size of exactly one. Just as in this aging clock that we saw, where you feed in all your biomarkers, your blood values, what if you fed that into the LLM instead? And then it would tell you.
That's what Nikhil actually was talking about digital twins.
Mm-hmm.
That you like you can feed specific information about you, your health, and so on, and things will get more personalized. Of course, like you need like general large data set to train some clocks, but for specific recommendation, you can pre-feed with your preferences. Also, not only health preferences, also, for example, if like even as general recommendation what sport to do, maybe generally it recommends, for example, running, and you don't like running.
Right. Just like so I want to make clear, I'm not trying to say that that using LLMs for anything where you have an individual result that you're trying to work on is a problem. So I'll have to go there. Okay. What I'm just trying to say is that you have to be very careful about how you use it. Because, for example, in your case, if you've got a few biomarkers and you want to put those in and use them for a specific individual case, you're going to base it on the statistics overall that you get from the population, because there are a lot of similarities between humans, of course, and how these individual components affect. Do I have to keep pressing it? Okay. Oh, okay. There we go. That's not a problem. And the same way, if you're using an LLM to deal with small components of the circuit, so if all you want to do is to reconstruct, for example, things that every human has, like we all have vasculature, and you need to reconstruct pieces of the vasculature that maybe weren't picked up well by a microscope or something, and you want to fix that piece of an image or something. Those sorts of things, fine, because these are things that we all share. It's when you get to the more complex things that are already put together in a very unique way, where if you then use an LLM to drive them towards the more general, and you're going away from the unique thing, then what happens are things like, oh, suddenly you no longer have in there the trained memory that mouse had, because now suddenly it's the general mouse, that sort of thing. So it's just an awareness of where to use it, really, or how to use it, being really aware of what your tool does.
Can we make a general claim about the suitability of LLMs to make personalized recommendations? Or is that something that will always be difficult? Yeah, I don't. I guess Milton. Okay.
Okay. Is there somebody wanting to react concerning the last interventions, or it's for something else? David, or you? Sorry, yeah, you have to come here. Sorry, so that the other people can also listen. And maybe if you want, you are not obliged, but you can present yourself in two words.
Hello, my name is Nicholas. I live in the Netherlands. You won't know me. I'm nobody at all in this field. I've come just purely out of personal interest. From a top down, I have a couple of questions I'm wondering about. First one is the motivation for this field of research. I can imagine altruism in it. I can imagine professional, commercial motivations, also personal motivations. I can imagine love of life, but also I can imagine fear of death. So that's two. I'm just wondering about the motivations in the entire field. The second thing I'm wondering is the impact on society, I think, should not be ignored. Obviously, from my point of view, if we suddenly gained enormous efficiency in this field and we extended everyone's life by 50 years, society would have a huge problem, especially if the extra 50 years were non-productive years. So this is an important consideration, I would think, that we should all also think about. So just a few 2 cents of mine I dropped in. Thank you.
Maybe this is more for tomorrow, because today is really a workshop about artificial intelligence and longevity. But I will answer that it is altruism, it is fear of death, it is hope of longer lives. But this is really the discussion for tomorrow, and also for tonight if you are tonight. But not. Yeah.
Personal recommendation issue.
Can I just add one more thing and then I'll hand you the microphone? The other thing, from my personal point of view, I would be interested in is not so much the prediction of the end of my life. I would be more interested in the prediction of the end of what I would define as my quality of life.
Yes.
So I would like to define my minimal quality of life that I would like to have to continue living. So I would like to define when I develop 3 cancers, that's the end. I don't want more life, or when I lose the ability to walk or talk, etc. So for me, that would be more important, predicting the reaching the end of that quality of life rather than the end of life itself. Thank you.
And then you want to know which drug is used to end that life.
Anton, you wanted to say something? Just also already to answer. So if you see at the entrance, it is not life, our organization HEALS, but at the others, it's not a life extension society, it is healthy life extension society. So to keep dying very, very long, yeah, it can be interesting for some people, but not for us.
Yeah, I want to address, so regarding OMG, I have to, to address personal like recommendations. So the main issue is that when like ChatGPT or any other model, when it makes mistake, it makes it in a very confident way. So it's you kind of get a result that is written in a nice style, and you think that the model knows what it's talking about, and it often is deceiving. And we actually played a bit with different personal recommendations. And for example, we had the experimental headache GPT just to have headache related experiments, and you have to be actually tweaking it in a proper way. For example, you have to push LLM to ask additional questions to tell how confident it is, also to self-verify, or you can use like maybe 2 LLMs to verify each other. So you have to use like different techniques to actually improve your results, because mistakes are often done in a way that is hard to catch intuitively like you do with a human. And another thing is that so far I mean, I would say that in the nearest term, the niche would be maybe when you don't have time or you have to wait a lot for an appointment with the doctor. So you use LLM like to get a quick answer. So I think doctors are still a bit more reliable right now. As models go better, it will change for the better. The thing is, I think many people are deceived by all those news telling like how well LLMs do medical exams, but the reason they are doing it well because of contamination that those exams students share them in the internet. So LLMs actually know many of the questions. That's why they do well on medical exams. But when you give it to real patient, they perform less, and also it is very field dependent. So in some medical fields it gives really good recommendation, but in other medical fields it fail miserably. So you have to be really careful.
Thank you, Anton. So great. Too many people asking to speak now. That's why I will give the word to myself. No, seriously, and then David, Charles, and you. But I would like to ask, especially to Anton and to the people who are online. So let's imagine that everything is going great, you know, and that we are working really for solutions for longevity, healthy longevity. Of course, when you are working on this, you are also working very efficiently to destroy life, you know, because it's the same way to have better health and to have worse health. So what to diminish the risk that this open science is bad. And I heard about something that, let's say, kind of an obligation for organizations to and for people to give an ID so that at least they can be followed if they are searching things. But again, that's one of the many IDs. So just asking this, Anton, if you have, or the people online, if you have directly an answer to this. Otherwise, I give the word to David. So about how to diminish the risk that AI plus longevity research is dangerous for people and not only useful.
Okay.
And you have to do this.
Okay, I see as others, I'm a bit afraid to be talking too much, but I think that what some large companies trying to do is to use AI risk for regulatory capture. Like they say, okay, let's regulate us, let's regulate all models beyond some capacity. And of course, they have a lot of money to have like red teaming, to have alignment and safety research. So one risk is a regulatory capture and the stagnation of industry because of it, and because of like overusing this issue with AI safety things. But at the same time, we also need some types of benchmarks and some transparency, because like to actually evaluate independently what LLM is doing. So and also to work more on explainability, especially not only on LLMs, but also on like regular aging clocks, to be, because the way I showed you like this 1 pixel attack, the way how AI reasons can be really very different from the way how we reason. And we have to push more on explainability research and to make things more transparent and traceable.
Thank you, David. David, then you, I don't know, and then Sven.
I'm like Anders, I came late, so I missed all the valuable discussion before. But I want to tell a story about my family. When I was young, one of my sisters fell with a disease, which in previous generations would have killed her. It was a disease that killed maybe 1 in 7 of all people who ever lived, including very many in childhood, disease of tuberculosis. But thankfully, in the time that I was growing up, there was a drug that would cure tuberculosis. So my motivation for the kind of work that's been described here is to allow more solutions like that to the diseases that are affecting people. My sister has lived, many decades since then, a wonderful life. But if that drug had not been available, she probably wouldn't have had that experience. So my motivation is to have more life, give more people more chance for more life. And it's important to keep our motivation clear. Nicholas is right that if we just follow technology for technology's sake, we'll often end up in a bad place. So I'm very motivated to find how technology can help us find new drugs. And recently, technology has suggested new drugs. I'm not sure if we talked about this before, but in silico medicine, a company in Hong Kong used artificial intelligence to come up with a new drug for IPF, which is idiopathic pulmonary fibrosis. It's a terrible disease of the lungs. It's called idiopathic, which means nobody's quite sure what causes it. And I know some friends who've got this disease and they're dying, sadly. I don't want them to die. They're full of ideas, but they may die in the next few years without a good solution. And it's defeated normal doctors to come up with a solution. And now this AI, this system from in silico medicine has come up with a potential solution. We don't know yet whether it's correct, and we have to test it. We have to push it through the phase 2 trials and so on. It's already past phase 1. It's the first time that an AI-generated drug has gone through phase 1 tests. I want to see more of that. But absolutely, we humans have got to test the results coming up. This is the risk that we run, that we might lose our critical capabilities. And AI might actually get the right result often. But then if we stop being thoughtful, it might give us another suggestion. And that suggestion might be like what? Thalidomide. Thalidomide was a drug in the 1950s that seemed to do a lot of good. People were glad to take thalidomide. And then it was discovered, oh my goodness, thalidomide is having terrible effects on pregnant mothers and on the children. And it took a long time to actually pin this down, and many people suffered as a result. And that tightened up the checking. And thank goodness we tightened up the checking. And with AI, as we heard, AI is often very good at coming up with a creative idea. It may suggest all kinds of things. But for the time being, we humans have got to use other methods to verify it. And the risk is that we'll get too excited, too carried away, too desirous of making profits, that we'll skimp on that testing phase. And we'll have something like thalidomide, but much worse. In other words, something that seems to cure people, that seems to make people stronger and healthier, but there are side effects. We already heard about a drug which had a side effect, rapamycin. We heard from Anton that rapamycin has the ability to make us live longer, but there is a drawback, which is that our immune system is suppressed and we may have this bad complication. And some people probably shouldn't take rapamycin because of their situation. So we do need to understand what the drawbacks are. So my view is that when we're using these LLMs in any of the ways that have been discussed, they can be wonderful creative partners. It's like a new intern in a company. A young new intern is full of bright ideas, but you don't let them go to the market with their ideas. You need a slightly more gray-haired person, maybe a few years older than them, to check whether their idea works. And so we humans, for the time being, need to check what these LLMs are saying. And it may be, as Anton was saying, that we can use one kind of AI to verify the results of another one. And this is what I think is going to happen a lot more in the next few years. We are going to see combination systems. After all, we humans, that's how we operate. We operate in teams like that. We've got creative people in teams, and we've got critical people in teams. And with that combination, we can come up with really good solutions. Inside our own human brains, we have a creative part. Sometimes with them, we have a type 2, the rational part that criticizes our own creative ideas. So I see the AI of the not too far future may be doing that. So that takes me to what I wanted to say when I put my hand up first of all, because I actually wanted to pass a question back to Randall, because Randall said, you know, he has these systems which can't do a certain thing. So have you talked, Randall, to some of the actual architects of these AI systems? And have you said, look, your system is good, but here's an issue it can't cope with? Because I suspect there are clever people inside some of these AI companies that may work in a combination system. But we here, none of us in this room, are deeply knowledgeable about the architectures of the systems that are coming. So have you actually shared your requirement? It's a good requirement. It's to deal with a special case rather than just a general case. Have you managed to talk to any of the providers of your LLMs about this solution?
We've spoken with people there, but it's not an issue of solving how making
But
LLM.
maybe-
It's about LLM versus a different method.
Just go there, yeah.
It's okay.
It's okay, okay.
Okay, thank you. That was just my point of view. I wanted to assert that this is a really good thing to do, but we have to be thoughtful and wise, otherwise we may regret the genie we're letting out of the bottle. I do think there are risks involved, but if we're smart, we can cope with the risks. Oh, can you hear me?
Yeah, but I don't know what.
Turn it on.
It's, yeah.
Hi, my name is Abhishek. I work in an organization called Vida Biotech in
I don't know what you.
Vida Biotech in Leiden, and I just want to suggest something. I don't know, because I would consider myself stupid in the field of AI because I'm not an IT person or anything. I'm just a biologist, but I'm trying to use AI. I remember seeing one saying in the door of a bioinformatician in Tübingen in Germany, where it said that if you screw enough with your data, you start singing what the song which you wanted to sing. Okay, so I know that more the raw the data, the better is the output, because more the process the data, you are mostly tuned towards giving the answer that it wants you to tell. So I don't know whether the questions, when you put it in the AI, on which database does it depend on to give you the answer. And I don't know whether you have a set of filter mechanisms to filter out sound or noise, which is coming along with that processing and giving you the answer, which takes you in a totally different direction or a wrong direction. Because the data that goes into a process is more important than the output. That is what I feel always, because being a biologist and an experimental biologist, I know that the data we put in is more important than the answer we get. We should not try to first, we should actually make a narrative after we see the data than making a narrative and try to make the data fit that narrative. That is my way of belief. So in that way, you won't have any kind of bias in the output. That is the first thing which I feel in most cases when I was using ChatGPT and trying to fine tune my question, first starting from a very general way of asking to coming to a more specific question, I found every time it gave me wrong answers. So I realized, okay, that means the data on which it is depending on to give me the answer is totally false. So I would also request you guys to please put a little bit of effort, not only on how to give the answer, but also to filter the data that you are taking in. Because I know out of 100% of publications that are there online in biology, 90% are stupid publications. I can give it in return. So I would like you to actually have a filter mechanism to filter out the noise and the crap. Because for example, for basic example I can give you, when we do mass spectrometry, we can only get 20 readouts. But that 20 readouts are the predominant readouts, but which we don't need. So we need to filter that out to get the minute readouts. The same thing when we do with next generation sequencing data. What we do, we get a lot of data, gigabase data, but until unless we annotate them and classify that data and put them in a compartmentalized form, we are not able to understand the data. So we need to compartmentalize the data before we rely on the data. That is my first suggestion. The second thing is to you, and I'm really impressed on the software that you're working on to read the functional readouts from the patient features and all those things. I don't know much about that, but don't you think it will serve a much more purpose if you also include occupational as markers? For example, a person who is working in a sulfur mine, he will have a different kind of readouts from his body compared to a person, because whenever you go to a big organization, the first thing a HR faces is burnouts. People are getting burned out. because so much of stress is given on them that at one point of time they go up to a burnout leaf. So if you are able to design, put the parameters which for specific occupations for an athlete, what are the symptoms of a concussion, or for a marathon runner, what is the stigmatism in his muscular tissue, or for example, for a person like me getting burned out by thinking too much. So the point is that if the software is designed for occupational purposes, then it will be a tool which the additional tool that a HR department can actually use it. Like, for example, if somebody is actually, if some things can be detected in their blood, or if something can be just detected on their body physical feature, that will be really helpful, because most of the times people lie to the HR when they are asked, are you okay? They will say yes, which is 99% lie. So if they are given additional tools to actually analyze the person and recognize the person before he goes on to burnout, to bring him out of that stress environment, then he'll be more productive than actually the person pushing him to the brink of burnout. Because I would say people do work long hours, but that is not productive. Literally not productive. Burned out persons produce nothing at all. So if it is you are having occupational markers, then that will be helpful for the people in real time. Then developing something which we can use it for our personal sake. So it will be really a good tool actually for management purposes, and also you can educate the management people to see how this can be used and what is the error possibility in that. Thank you.
Okay, thank you. So there are two people who asked the word for this first part, and after that I would like to have the last part of this working group. And a reminder, the last part is about attempts to draw a first concrete picture list of possible improvements, collaboration, and changes. And I propose that you are thinking this way. So what if I had, or if we had $1 million, 1 million, not 1 billion, and two full-time people, full-time scientists, one for IT and one for longevity science. And the same question maybe with 1 billion and 1,000 people. Yeah, so welcome to your ideas, but first there was yeah. And I propose now on maximum 2 minutes, sorry, the last one, sorry.
Yeah, mine is pretty simple. I'm not so quick at part of all this LLMs and what have you. But the tools that we were given to play with, like reading your face or, reading blood samples, and I hear that we had to enter like name and age and what have you. So I'm worried about privacy and security. What happens with this data? Is that protected? Have you guys thought about that?
Okay. You can begin, and then I will give one sentence, and or maybe after Sven, and we will answer maybe. Yeah, Sven. And you can begin to answer.
So I wanted to come back to a question that Didier asked, and Didier asked, is there a risk to longevity GPT? And my answer to that is probably not. So I agree that there are risks to AI in general. So for example, you could make deep fakes, and you could create division between people by having a certain group of people deep faked and say things that cause problems and things like that. But when it comes to longevity, I don't really see much risk. If you wanted to kill people, there are lots of ways already to do that. So you don't need to invent a drug that accelerates aging for achieving that. You have acute poisons and chronic poisons, so you can achieve that already. Maybe the only thing I could see as a potential risk is that an AI tool that could predict mortality or morbidity will be misused by, for example, insurance companies to screen out people and say we are not going to insure that person because they are going to get sick in the future. That will be, I think, the only big risk to longevity AI, and that's one that I think regulatory agencies can address relatively easily. Thank you.
It's not just insurance, also banks, when you want to have a loan.
Anton for the answer, but yeah, go ahead and then I will also.
Yeah, so I first so first you like address like the clocks example of which I showed, it was from different sources. So for the video clock, it was developed by Andrei Batescu and his team, while the clock for blood, it is developed by Nick Hill. So far, in both cases, there is like general GDPR regulations for European Union, and for Nick Hill, I think it's HIPAA because it's US based. I think the web version actually is more like a research tool that he showed, and I don't think that web version actually saved anything. The Android app, it's probably saves, but the thing is, you should probably directly contact Nick Hill and contact Andrei to clarify how exactly things are used. Also, I can mention that there are ways to do machine learning on data without knowing the data. So this is called privacy preserving AI, and there are like many methods, including zero knowledge proof based and including hypomorphic encryption. Usually they get more resources and get worse results. But for like privacy crazy people, they actually do the job. I myself, I mean, for example, for things that I develop, I like for just DNA-seq project, for example, there we provide everything to run everything on your laptop. So if you don't have viruses, probably you are the one in control of it. I personally open sourced my genome, so I don't worry much, but you can run stuff on, for genomics, you can run stuff on your laptop. So it's everything, it depends on like a specific service that you are using.
And yeah, concerning all these the questions around privacy, I will speak about that tomorrow also, and I can speak privately, but let's be short. First aspect is when you ask people, do you want to share your data for scientific reasons? They say in a very, very, very large majority, yes, as long as insurers and so on don't know about my data. And at the European level, there is a regulation. Well, at the moment, it's a project of regulation called the European Health Data Space. And one of the basic principles there is people at the European level can share their data with their medical doctors and also with scientists, but they can, the insurance and nobody is allowed to use it for another goal than scientific data, the scientific goal, or than a goal in concerning the health. So this is really the point about this. And just a few words, weeks ago, there was a vote in the European Parliament, and there was a very large majority in favor of the text that is now in discussions for what this is called, that's very technical, the trilogue. So if everything is going great, in 1 or 2 years, there will be a regulation, in my opinion, about perfect. Nino, you want to say something? You have. And then after that, it's 20 minutes left before the conclusion. We will need to proposals of things. Right.
Yeah, just one last thing. A big part of the EHDS project is about anonymization of data for scientific use. So yeah, normally there is no problem with insurance, etc.
Anonymization and pseudonymization are two parts. And now, so the word to you, but be careful. It's not about questions. It's about kind of, well, ideas already. Yeah. Try.
I can try, yeah. So maybe this might be obvious, but when we're thinking of things like privatization and anonymization, I think the other part of this is also making sure that the data is actually verifiably correct. Because especially if you think it's going to be used for various other things, like seeing if you're going to be good for the athletic team of a school or something like that and get a good grant that way or something, get a scholarship. If it's going to be used for anything, there's always going to be a tendency for people to want to cheat the system. Everybody's going to want to game the system. As long as we're typing in the numbers ourselves, that's very easy to do. So it would have to come straight from the blood test or something like that. So in addition to anonymizing, privatizing, you probably want some sort of way to make absolutely sure that these are the correct data, maybe some kind of a hash or something. I don't know. Yeah.
Yeah, I actually agree with this option. I also wanted to add one more thing is that, for example, for aging research, one of the issues is that we pretty much need everything. Like usually in most like narrow use cases, you can limit the markers that you need, but for aging, because it's about most of our health and physical indicators, even social stuff, as we know, like people who are maintaining tight social connections or live with the partners, they live longer. So there it's actually really hard to, if we go hard on with privacy, it will be a bit hard for aging research. At the same time, as it was mentioned, there are technologies that, like zero knowledge proofs, for example, you can make a proof, like in your case, like if you have a zero knowledge proof that it's like some data verified by some source, you can do it without actually providing the data. And the thing is that for zero proof implementation, it's a bit technical, but if we really want it, we can actually think the architecture that will minimize data sharing and will maximize provenance of some properties about the data.
Now, who is wanting to say things about proposals, ideas, and so on? If nobody is beginning, I will begin to say, but for me, it's clear. So I really think that the work that Anton and others are doing is great. Of course, one of the first important things is to have access to many more scientific articles related to longevity than it is the case at the moment. And one big problem is the problem of what's called curation of data. And also it's the same for the as fast and as far as possible to have access to all health data who is really already publicly available. Also there with the big problem of curation. And that's what I want to say, but I don't know about you. If nobody is going to speak, I will give the word. David, and maybe you, because you were not speaking yet.
Yeah, before I ask, would you mind clarifying what you asked earlier about the $1 million and the $1 billion? What was your question at that point?
So my question is, I propose to think in two directions. You know, a small budget of 1 million. You have $1 million. What would we do? $1 million and one scientist IT and one scientist longevity science, let's say. And then the same question, but with a very large budget, almost without limit, or at $1 billion, you know, the famous $1 billion that Evolution was going to give to longevity science, and that they forgot to give. Okay. What was $1 billion a year?
Yeah. I'm not sure if what I'm about to say addresses what Didier mentioned. But what I want to ask you is, who here drives a car or a vehicle of any sort? If I get a quick hands up. Okay, most of us here. the beautiful thing about a car and a vehicle is we have a dashboard, right? You can see when you're going too fast, too slow, when the fuel is empty, whether there's something wrong with the car. Now, what I love is I wear a wearable, and I think it's good enough. It's not perfect, but it is good enough, and it gives the indications of when I'm going off track and when I'm not. However, the trouble with it is it's primarily non-invasive tracking and measurements. So the question I have, or the proposal is, some of these biomarkers you're talking about, and now we have AI, but I'm an artificial idiot. I don't know if we can actually physically measure some of these molecules in our blood non-invasively in a way which is very accessible. Because what I'm afraid of is we will let perfection be the enemy of good. So something worth doing, we won't do it because we can't perfect it, right? So I guess the $1 million or the $1 billion project would be, can we measure some of these molecules in the blood? Is it an engineering problem, or is it physically impossible to do that, to really get to that great dashboard? I'm wearing this Whoop. I've seen other people got Apple Watches on, the rest of it. Can we improve that? Can we get that to a level where it's very accessible for people?
Thank you, David.
I'll give my real answer to this question tomorrow afternoon when I'm giving a talk on getting to longevity escape velocity sooner. But there is something that badly needs money just now. There's a project that's been cancelled, which is criminal in my view. It's the dog aging project by Matt Kaberlein from the University of Washington. He is several years into a project to give rapamycin to middle-aged dogs and find out how the dogs are responding to it. And there are lots of interesting results. And we're beginning to find more evidence that if you give this drug in a suitable quantity, your middle-aged dog has a new lease of life, a lot more lease of life. And it's almost at the point when the results are available, and it's going to cause people to say, my goodness, this is something that needs a heck of a lot more attention. It's real-world longevity. And guess what? The funding has been withdrawn for reasons that are not clear at all. The FDA, it's not quite the FDA, I'm sorry, the NIH, the National Institute of Health in America, has decided that this isn't close enough to human health. It's not something that justifies. And there are all these dogs that are partway through the treatment, and the funding is going to be withdrawn. They won't get any more. We won't be able to draw proper statistical tests results. So that's one thing I would do if I had a few million. I'm not sure what the budget is that the NIH has decided it's a poor use of time, but the people who are closely involved in this project are really distraught. As part of showing the world that treatments can make a big impact, because a lot of people in a certain part of their life, they love their dogs more than they love their spouses. If you ask a woman or a man of a certain age, you've got to choose, get rid of your pet dog or get rid of your husband or wife, they'll say, you know what, I'd rather have my pet dog. Sad to say, it's a bit about contemporary marriage perhaps, but anyway, if we can allow this short-term impact, because you don't have to wait 80 years to find out whether this drug is going to have a result, a big impact, because dogs typically live just 10 years. So all we need is a few more years of this, and it'll be clear a lot more what the rapamycin can do. And when it's clear that it works on dogs, it's going to make the attitude of many, many more people around the country say, let's put more funding into this. So that's one thing I'd spend money on. Another thing I'd spend money on goes back to a point that was just mentioned, that we need to clean up the data. Actually, I think we need to do something more drastic than that. We have to clean up the scientific literature. Because all the papers that are published and which are being fed into these large language models, so many of them are poor quality. They're being driven by the wrong incentives. More and more people are encouraged to publish at all costs, and a lot of the papers that are published actually can't be replicated. There's a replication crisis. And so it's no wonder if you feed all this stuff into large language models, it's not going to get the right understanding. And so the incentives under which academics are driven to publish at all costs and to get as many publications on their CV as possible, it's really detrimental to the well-being of science. So that's probably going to cost more than a few $1 million to fix. We have to encourage a higher quality of publication, and Ben Goldacre wrote about it a few years ago in his wonderful book, Bad Science. We know the solution, we need to be more vigorous in pushing it through. If we do that, then we're more likely to have AIs that are not misled by lots of junk science, and they'll be quicker to come to the solution. So there's two things I would spend money on, but the most important thing to spend money on, I'll tell you that tomorrow evening.
Thank you, David. Well, one part of the answer was more about longevity in general, but about longevity and AI. So if I had 1 billion, I would give it, of course, to you and Aubrey for the Longevity Escape Velocity Foundation for the experiment on mice. But this is maybe another question. But also going further concerning what you said, that was one of the things that I was planning to say. Curation is especially important in the fact that at the moment there is no obligation, or no, in many cases, no obligation to publish when you have negative results. You know, like there are many people saying that metformin is not working for longevity, but no publication about this because of many reasons. Because it's of course more difficult to say, oh, I was thinking this was going to work and it's not going to work, than saying, great, I found something. That's one. And then maybe still, if I had not $1 billion, but if I had 1,000 people working full time for something other than really science, I would ask them to be very, very active to try to convince the DARPA for health, ARPA-H in the United States and the European health data space to work really on AI for longevity. And if you really push people, you know, you probably even don't need 1,000 people to be efficient enough to make people change their mind. As far well, sometimes you make them change their mind in a bad direction if you do that too much, but that's another question. So I propose now that you have about 30 seconds to raise the hand, each of you. If you don't, it will be the word only to speak about the next part and the real part of TransVision. And there is no 15 seconds, but before, maybe Anders, no other comment or one sentence? No, nothing? Okay. That's for later. Okay. The last person is here. Maybe you can also present yourself in 1 sentence, and then you give the word to
Hi, Cyril, I'm following AFT. So first, sorry for my French accent, and sorry if I do a panic attack for speaking in public. Actually, I have one comment about what you said about risk for Longevity GPT. I don't agree, because there is a risk. You said that, okay, we have better means to kill one people in particular, but you can actually poison data. Imagine Longevity GPT for now is speaking only English, but like ChatGPT can have more languages. And in English or other languages, Longevity GPT will say, don't smoke, don't drink alcohol, it will reduce your life. But you can, some state, any state can poison data, and in French, for instance, British will be happy. In French, say, contrary, oh yes, you must smoke, you must drink, because it will help you. And so you can genocide. So I take a French as an example, but some country could try to poison data in order to genocide another. Today, it's a case with some weapons. We have 2 or 3 examples right now, but you can do that through Longevity GPT. There are risks. Because I took French in example, some French people don't speak English. We have an example in that room, for instance. So yeah, there are risks, I think.
Thank you. Concerning risk, we will continue also to discuss. I'm almost sure that there is one speech about this tomorrow or Sunday, I don't know anymore. But now the floor for the conclusion is to Charles, and this precisely at 5:45.
Thank you, Didier. Let me just get set up here.
Maybe you should connect on Zoom because for the Zoom people, they will not be able to see the.
Ah, that's probably that's.
All right, I'll keep this brief. I'll just make the announcement for sessions tomorrow. Today was the satellite workshop of TransVisions, where we spoke specifically about longevity and longevity GPT. So thanks all for the very interesting discussion and input. I'll just briefly announce the program for the next 2 days and invite you to, if you're still considering to do a lightning talk tomorrow, we're going to have a time slot of half an hour just before lunch, where everybody will get the opportunity to present any topic of their choosing in an elevator pitch style presentation of 3 minutes. You're very welcome to still sign up. We have some free spots. So I made this link to it, sorry, no QR code, very old fashioned, URL/TV Lightning. You can sign up there and upload your slides. Tomorrow we start at 9 in the morning. Looking forward to seeing you all there. And just as a final note, this is the official end of the session today. But unofficially, we're going to continue over beers and drinks. And we're going to do that in a pub called Floden, which is just 11 minutes walk from here. You can also find the name and the address on our website, TransVision-conference.org. And we look forward to seeing you there from about 7 o'clock. DJ, do you have any final words to close today? Any final words?
No, thank you for being here. And yeah, don't. Thank you for being here. Don't be there on time, because it's not like in France, we live in more or less on time.
Strictly at 9 o'clock.
Straight 9 a.m. precisely, and we are more than open to have questions and discussions later. See you tomorrow morning or tonight. Okay.